So thank you, Christopher, and thank you everybody for taking the time today to listen to my presentation. So today I'm going to talk about this Oncogene GOLPH3, that is a PI4P binding protein, how it regulates the sphingolipid metabolism and cell proliferation. So here you can see how the secretory pathway is organized in mammalian cells. So protein and lipids that need to follow the secretory routes are first synthesized here at the level of endoplasmic reticulum, and then they reach the Golgi, where during the transport from the cis site to the trans site of the Golgi, protein and lipid are fully decorated with sugar moiety through a process that is called glycosylation. This process is exerted by glycosyl transferase and glycosidase, and then finally, the cargo, when it's fully processed at the TGN level, is sorted to the final destination, the plasma membrane or the endolysosomal system. So here you can see some example of the glycosylation exerted by the Golgi complex. You can see N-glycosylation, glycosaminoglycans, hyaluronic acid, or glycosphingolipids, for example. And this modification affects solubility of protein and lipids, localization, half-life, and I will say cell signaling and many other functions. So this is a complex process, and while in the case of DNA, for example, we have a template where starting from a genome information, we can predict which will be the final output, meaning a peptide or a protein. In the case of glycosylation, this biosynthesis of glycans is not based on a template. So the situation here is a bit more complex, because starting from a genome information, the output will be stochastic. So that's the reason why understanding how Golgi reads and translates the genomic information in a faithful glycan output is not clear. So there are mainly a couple of, I will say, two main factors that contribute to the glycosylation. From one side, the expression patterns and catalytic properties of glycoenzymes, and this is the more studied aspect, I will say. But less is known about the localization of glycoenzyme at the Golgi and their regulation. That is what I'm going to talk about today. So during my PhD, I was mainly trying to understand whether Golgi enzymes are static and fixed within stable cisterna, or whether these enzymes are dynamically recycled back using COP1 vesicle in order to be localized in the right cisterna during the anterograde movement of the cisterna, what we call cisterna maturation. To test this hypothesis, I took a cis-Golgi enzyme, and then upon polymerization of this enzyme in a specific manner, I was able to impede the entry of this enzyme in retrograde carriers, and after that, I observed that the enzyme in the time of intra-Golgi transport moved from the cis site of the Golgi to the trans site of the Golgi in a cargo-like fashion. But more interestingly, upon the polymerization, the enzyme very fast, in a couple of minutes, enter again back in retrograde carriers to start to relocalize in the cis medial place. So this can be simply summarized, I would say, by this animation, where you can see that cisterna move forward to transport the cargo, here represented by black dots, while Golgi enzyme with different colors always maintain the same position thanks to this recycling process. So, but this was the first demonstration in mammalian cells that the enzyme recycled. But which is the molecular mechanism behind this process is not known. So starting from this question, we use as a molecular system, as an experimental tool, I will say this GOLPH3. GOLPH3, that is a PI4P binding protein, is also an oncogene and works also as an adapter that binds Golgi enzyme. At the same time, GOLPH3 can be easily manipulated to alter the intra-Golgi dynamics of its clients and possibly glycan output and cell growth. We went back to the literature and we find that the same protein in yeast is called BPS74. And what has been shown by Banfield and co-worker is that a subset of enzyme that normally localize at the Golgi, upon depletion of BPS74, they leave the Golgi and move to the vacuole where they are degraded. While upon overexpression, the same enzyme move back to the endoplasmic reticulum. So the model proposed by Banfield and co-worker is that GOLPH3, which is recruited at the Golgi thanks to the binding with PI4P, from one side binds COP1, and from the other side, it binds the cytosolic tail of a subset of Golgi enzyme. By doing so, GOLPH3 incorporates this enzyme in retrograde COP1 vesicle, thus allowing this retention of this Golgi enzyme in this organelle via a recycling mechanism. Not all the enzymes are clients of VPS74 in yeast, but just a subset of them that contains in their cytosolic tail a specific sequence of amino acids. I will say a motif, okay? Starting from this information, we actually realized that in mammalian cells, there is a similar motif, but is a shorter version, that is also present in several Golgi enzymes. We cluster all these enzymes and we find out that this shorter version of the motif, that is LXXRK, is mainly enriched in the glycosphingolipid biosynthetic enzyme. And here on the right, you can see a very simplified version of the pathway, where ceramide, that is the main precursor, can be converted in sphingomyelin thanks to the activity of sphingomyelin synthase 1, or can be further glycosylated by glucosyl ceramide synthase in order to produce glucosyl ceramide. Then glucosyl ceramide by lactosyl ceramide synthase is converted to lactosyl ceramide that will give rise then to globoside, ganglioside, lactoside, and asialo, that are the complex glycosphingolipids. So since GOLPH3 is an oncogene and when it's overexpressed, when it's amplified, the first things we have done is to overexpress this oncogene in cells and check whether there is any change in sugars on the membrane. So for this reason, we have done lectin screenings, and with our surprise, find out that for whatever reason, GOLPH3 overexpressed in cells binds better Shiga toxin, which uses a receptor GB3, the globoside GB3. After that, we decide to find out which are all the interactors of GOLPH3, all the Golgi glycosyl transferases in the glycosphingolipid biosynthetic pathway that are clients of GOLPH3. So for this reason, we use this assay that are avidin beads that were functionalized with biotinylated cytosolic tail of the main glycosphingolipid biosynthetic enzymes. These functionalized beads were used as a bait to pull down GOLPH3 from total cell lysate or using the purified proteins. So to cut the long story short, what we have found is that GOLPH3 directly binds GD3 synthase, lactosyl ceramide synthase, GM3 synthase, and GB3 synthase. And this binding is direct. Next, we ask whether the information to retain this enzyme in the Golgi is located in the cytosolic tail. For this reason, we use an experimental system, the sucralose maltase, that is also a type 2 membrane protein, which normally cross the Golgi and localize to the membrane. So if you both transfect lactosyl ceramide synthase and sucralose maltase in the cell, while the enzyme localizes in the Golgi as expected, the sucralose maltase cross the Golgi and move to the plasma membrane. So using this system, we just swap the tail. So meaning that we implant all the cytosolic tail of the GOLPH3 clients on top of the sucralose maltase and test where this chimera localizes. As you can see in this experiment, while sucralose maltase localized in the plasma membrane as expected, in the case of the chimera containing the tails of lactosyl ceramide synthase, GB3 synthase, GM3 synthase, and GD3 synthase, all these chimera localized at the Golgi, meaning that the information to keep the enzyme in the Golgi is mainly in the cytosolic tails. More interestingly, if you remove GOLPH3, all these chimera leave the Golgi and move back to the plasma membrane, behaving like the normal sucralose maltase. So GOLPH3, the information to keep the enzyme in the Golgi is in the tails and require the binding to GOLPH3. So then, to go deep inside in the, which are the amino acids in the cytosolic tail of LCS that are responsible to the interaction with GOLPH3, we actually use the same biotinylated assay, the biotinylated beads with the mutated cytosolic tails of lactosyl ceramide synthase to test the interaction, and also mutated tails of the sucralose maltase, meaning that we implant on top of the sucralose maltase mutated tails of lactosyl ceramide synthase. Anyway, to cut the long story short, what we have found is that GOLPH3, in order to interact with lactosyl ceramide synthase, require the binding not only with the LLXRK motif, but also with the pool of arginine that are in the most N-terminal part of the enzyme. So interestingly, all the enzymes that are clients of GOLPH3, they share this dual GOLPH3 binding motif. So next, to go deep inside the GOLPH3 LCS binding mechanism, we use rigid body docking. Initially, with our surprise, we found out that the clients of GOLPH3, they organize, at least in silico, their cytosolic tail in an alpha helix. These enzymes were docked with the crystal structure of GOLPH3 in order to identify which are the amino acids that contribute to interact, to participate in the interaction with the cytosolic tail of LCS and all the other GOLPH3 clients. So these amino acids that were found out with this rigid body docking were mutated in alanine, and after mutating this amino acid in alanine, we can abolish the interaction between lactosyl ceramide synthase and GOLPH3. So, and here the first summary. So now we know that GOLPH3 binds and retains at the Golgi a set of glycosphingolipid biosynthetic enzyme by interacting with the structural motif in their cytosolic tails. Next, we decide to study more in detail the localization of the enzyme. So if you see the enzymes that are deprived of the dual GOLPH3 binding motif, like sphingomyelin synthase 1, glucosyl ceramide synthase, LC3 synthase, and DM2 synthase, they are all insensitive to GOLPH3 expression level, meaning that they normally localize at the Golgi, and they remains there even upon knockdown or overexpression of GOLPH3. While if you look at the enzymes that are clients of GOLPH3, they normally reside in the Golgi, but upon GOLPH3 knockdown, they leave the Golgi and appear in cytosolic puncta. While upon overexpression, the same subset of enzyme move back to the endoplasmic reticulum. That is a similar phenotype that was observed in yeast. So at this point, since lactosyl ceramide synthase is the strongest interactors and also is the most upstream enzyme in the glycosphingolipid biosynthetic pathway, we focus our attention on this enzyme and we look at the endogenous lactosyl ceramide synthase. Upon GOLPH3 overexpression, the fluorescence of the enzyme increases in the Golgi, while completely disappear upon GOLPH3 knockdown. This change in fluorescence also corresponds to a change in the levels of the protein, meaning that GOLPH3 positively controls the level of lactosyl ceramide synthase and also all the other GOLPH3 clients. So next, we decide to check more in detail, which is the trafficking step in which GOLPH3 is involved in the retention of lactosyl ceramide synthase. So we use a synchronizable version of this enzyme, where we keep the enzyme in the AR and then we release the block and check what happens in GOLPH3 knockdown cells versus control cells or GOLPH3 overexpressing cells. As you can see here, starting from the right and going to the left, in all the cases, the enzyme moves from the ER and reach the Golgi, but in GOLPH3 knockdown cells leave the Golgi in the absence of the adaptors and move to this spot, while in GOLPH3 overexpressing cells, the time spent by the enzyme in the Golgi is long, till 10 hours of acquisition. And this can also be followed biochemically. It's also interesting to note that even in control cells, there is a leakage of this enzyme to this puncta. This spot lysosome, as you can see in this experiment, and as can be emphasized by the use of bafilomycin, that is an inhibitor of lysosomal degradation. So the enzyme upon GOLPH3 knockdown appear in this spot, that are lysosome, while the normal leakage of the enzyme that is observed also in control cells, it's completely abolished upon GOLPH3 overexpression. And the same experiment can also be followed biochemically. So, going more in details at the ultrastructural level to see where the enzyme localizes in the Golgi, we did cryoimmunolabeling, and as you can see in this cartoon that simplifies all the experiment, in control cells, lactosyl ceramide synthase, here indicated with red dot, is normally in the transcisterna and also appear in COP1 coded vesicle, that are these retrograde carriers. Upon GOLPH3 overexpression, the rate of recycling, the internalization of the enzyme in this round profile, it's increased, I will say, and the enzyme not only increases in the Golgi, but also appear in the cis-medial site of the Golgi. So this recycling is accelerated upon GOLPH3 overexpression. If you see in knockdown cells, the Golgi is almost empty, meaning that the enzyme move from the cis site to the Golgi, from the cis site of the Golgi to the trans site like a normal cargo, but in the absence of the adaptor cannot be retained and recycled back. That's why leave the Golgi, reach the TGN before localizing in the lysosome where it's degraded. So here, just a cartoon that schematizes whatever I tell you so far. So GOLPH3 mainly acts as a rheostat that controls the flow of its clients to the lysosome, mainly lactosylceramide synthase. So, and here the second summary. Now we can say that GOLPH3 controls the protein level and localization of sphingolipid glycosylation enzyme, bringing the dual GOLPH3 binding motif. This happened at the Golgi. But what about the pathway? Because so far we have seen how the enzyme of the glycosphingolipid pathway behave. So in order to check the metabolism, we use several approaches. We use Sphingosyn pulse chase experiment followed by lipid extraction and chromatography. Then we have done mass spec of the lipid following mainly ceramide, sphingomyelin, glucosylceramide, lactosylceramide, and also GB3. And then we monitor also using Shiga and cholera toxin the level of GB3 and GM1 using immunofluorescence as well as FACS analysis. All these results together suggest that upon GOLPH3 knockdown, since we lose the enzyme, we have an increase of ceramide and glucosylceramide, while then the glycosphingolipid downstream to glucosylceramide, they are lost, they decrease. The other way around, if you overexpress GOLPH3, the enzyme increase, mainly lactosylceramide synthase, and this gives rise to a consumption of ceramide with the consequence increase of GB3, that is the main glycosphingolipid present in these cells. So now we also know that GOLPH3 positively control the levels of glycosphingolipids downstream to glucosylceramide. So GOLPH3, I will say, is at the center in this slide, where from one side control the trafficking of lactosylceramide synthase and all the other glycosphingolipid biosynthetic enzyme, and through this trafficking process, it actually regulates and I will say reprograms the glycosphingolipid biosynthetic pathway, thus causing, when it's overexpressed, a consumption of ceramide, that is a growth arresting compound, with the consequent increase of GB3. So at the beginning, I told you that GOLPH3, that is a PI4P binding protein, is also an oncogene, right? So this was shown initially and proved by Scott and Chin, which actually demonstrate that GOLPH3, when it's amplified, it causes the proliferation via the activation of mTOR signaling pathway. So we ask whether glycosphingolipids are involved in this process. For this reason, we monitor at the same time GOLPH3 and also lactosylceramide synthase, his main interactors. And we find out that both proteins, GOLPH3 and LCS, when they are overexpressed, promote proliferation as measured by soft agar colony formation, while the growth is affected upon GOLPH3 or lactosylceramide synthase knockdown. Both proteins also mimic each other, I mean, they phenocopy each other, at least looking at the mTOR signaling pathway, meaning that they both activate mTOR or they switch off the signaling pathway upon their knockdown. So to test the dependency and check whether the activity of GOLPH3 on mTOR and growth depend on lactosylceramide synthase, use knockout cell line for the enzyme lactosylceramide synthase, but also the glucosylceramide synthase, the enzyme before. Very interestingly, the mTOR signaling pathway that is activated by GOLPH3 is not active anymore upon LCS knockout, I mean the downregulation, meaning that GOLPH3 need LCS to activate mTOR. This is true not only in signaling, but also in terms of growth, because if you take knockout cell line for lactosylceramide synthase and you give GOLPH3, nothing happens to proliferation. But in order to restore proliferation in this cell line, you need to give back lactosylceramide synthase or another oncogene like RAS, valin 12. So whatever I told you in this experiment can be simplified by this cartoon on the left side, where GOLPH3 through lactosylceramide synthase promote the production of glycosphingolipid that via mTOR1 and mTOR2, they promote proliferation. So whatever I show you so far is just experiment done in the lab, on the bench, using cell line. So, but to test whether this is true also in a real life, we took more than 70 patients, 70 biopsy of patients with human lung cancer. We cut the section and stained for GOLPH3. And we find out that when there is a lot of GOLPH3 protein-wise, there is also a lot of lactosylceramide synthase. So both GOLPH3 and LCS levels correlate in human lung cancer. And this can be easily explained by the mechanism that I show you so far. So now we also know that GOLPH3 promote mTOR signaling, impacting on glycosphingolipid levels. I think I almost can. So I just want to give you some message. So the take-home message here is that from one side, GOLPH3 acts as an integral component of systemic maturation, meaning it recycles in the Golgi a subset of Golgi enzyme. This suggests that at the same time, there could be other adapters that binds other subset of enzyme in order to promote the recycling during the systemic maturation. Then, second message is that GOLPH3 acts as a rheostat that controls the flow of its clients to the lysosome. And this is interesting because this is a novel lysosomal degradation-based regulatory process to control glycosylation. And finally, GOLPH3 alters the glycan pattern assembly on glycosphingolipids to produce more proliferating inducing glycosphingolipid metabolites, in this case, mainly GB3. To put all this data together in a more general context, I will say, here the main player will be the PI4P, that is a master regulator of lipid transition at the Golgi. PI4P, in fact, it's required for the transfer of ceramide from the ER to the Golgi, thanks to the activity of SERT. Then we have OSBP, which transfers cholesterol from the ER to the Golgi. Then there is FAP2, which transfers glucosylceramide from the C site of the Golgi to the trans site of the Golgi. And now entering the game GOLPH3, which through this activity and the binding of this enzyme, it will position this enzyme at the trans site of the Golgi, thus digesting this glucosylceramide in order to produce lactosylceramide and all the other glycosphingolipids. And here, the most important slide. So I have to thank several people that contribute to this work, but a special thanks need to be given to these two people on top, my boss Alberto Luini and Giovanni D'Angelo for the overall direction of the project. And finally, thanks to all of you for your attention. So please, if you have any questions, I'm here.